Our first speaker is Dr. Stephanie Langel from Duke University. She's a medical instructor there. Stephanie's background is she got her PhD at the Ohio State University in 2018 and has been a postdoc at Duke and is now a medical instructor there. And her research focuses on understanding maternal and neonatal immune responses in order to prevent neonatal viral infections. So she's very familiar with viruses in the human body and is making tremendous contributions there. She's also a host of the podcast Immune, a monthly podcast about host defense systems that protect against disease. So Stephanie, thanks for being with us today. We'll get you to jump right in.
Great. Thank you so much, Dr. Jordan. I really appreciate the invitation to speak today at the American Peanut and Education Society's annual conference. I appreciate that everyone is virtual this year. I know that is challenging, and thank you to the organizers because it is a lot of work to put on a virtual conference. So I'm going to talk today, as Dr. Jordan mentioned, about coronaviruses in the human body. I do come from Duke University, was at the Duke Human Vaccine Institute, and now faculty in the Duke Center for Human Systems and Immunology. Working on viral immunology, was working on HIV, rotavirus, other viruses than SARS-CoV-2. And then, like many of us did, switch to do SARS-CoV-2 research once the pandemic hit. So I'll be happy to answer any specific questions at the end. But the goal of my talk today is to go through what coronaviruses are, what is the pathobiology, so what is happening inside the body of a human as it reacts to the virus binding to the receptors on our cells and making millions and billions of copies of itself, what is the trajectory of disease, and then followed by the immune responses and how that relates to both natural infection and then after vaccination. And now we're in this post-vaccine world, and what does that look like in regards to viral variants looking to the future? As we enter into another cold and flu season, where what we know about SARS-CoV-2 is it is spread by respiratory droplets and is really potentiated by being indoors and close to each other. So as we all come back together inside as it gets colder, what are the implications for now a post-vaccine world? And just a little bit of background, I do feel a little bit at home. I did come from an agricultural background. I milked cows growing up and was in 4-H, and I did my PhD in veterinary coronavirology. So those of us in this field have known for many, many decades that coronaviruses have vast pandemic potential, most well known by the pork industry, which is what I did my research in, was PEDV, or porcine epidemic diarrhea virus, which emerged from China in 2013 and killed about 10% of the pork population. And so after that, coming to Duke and really merging my interests in human and animal interactions of viruses, which really could be no more relevant than today when we talk about SARS-CoV-2. So I'm really excited to present to you and look forward to your questions. So what are coronaviruses? Coronaviruses are in the Coronaviridae family. They come in 4 genera based on sequence similarity or phylogeny, and those genera are alpha, beta, gamma, and delta. These are enveloped viruses. They are positive sense, which just is a way that they replicate, is pretty unique, and they're single-stranded RNA. And because they're RNA, they're vulnerable and they need to be protected by the nucleocapsid protein, which you see kind of squiggly inside of the virion of this virus. The membrane really helps to anchor the envelope proteins, which are shown here. And then the spike proteins, which likely you've heard of quite a bit, are maybe considered the most important protein of this virus because they are the antigenic determinants of this virus. They drive virus neutralizing antibodies, and they are what has been used in the vaccine because they bind the receptor on our cells. And so if you can block the binding of this virus via its spike proteins to the receptor, you could potentially help block or limit spread of infection. So coronaviruses, this is not the first coronavirus pandemic or epidemic that we have been hit with as humans. So I mentioned there's been many in animals as well, but I just wanted to talk about the 3 emerging coronaviruses in the last 20 or so years. So the very first coronavirus that emerged was severe acute respiratory syndrome coronavirus 1. We didn't call it 1 back then, but SARS-CoV. It emerged in Guangdong, China from civets and raccoon dogs. It was later determined by sequence homology of the viruses that were recovered from those animals. And one theme you're going to hear a lot, and really was touched great upon Dr. Widmer's presentation before me is this interaction of humans and animals, humans and the environment, humans and agriculture, because coronaviruses are zoonotic pathogens and they have a huge propensity to jump species because of their ability, we think because of the replication strategy. So in 2002, this virus emerged, and within less than a year, it was really contained. It killed about 772 people. It's 10% of the people who were confirmed infected. And there's a reason for this kind of quick containment. And that's because the time to onset of disease was 2 to 7 days. People got quite sick quite quickly. And you can imagine if you're feeling crappy, you're going to end up staying home or you're going to go to the hospital to get treatment. So you can contain the vector, which is the human spreading virus out into the environment, because we can get them in a hospital, get them home. And essentially this virus we were able to contain it because the hosts, the humans, we were able to limit spread. And so you can imagine from an economic perspective, there wasn't much motivation to make a global coronavirus or a universal coronavirus vaccine, because the virus really never made it far. 29 countries, the US was not one of them, which is where the major vaccine manufacturers are. So when we talk about, well, why wasn't there a vaccine for SARS-CoV-2? Those of us in the field would have loved that. And we wrote that in our grants. These are viruses with pandemic potential, but really there was no motivation back in 2003 to do that. About 10 years later, MERS emerged, and this is mostly in the Middle East, first detected in Saudi Arabia, and it's from dromedary camels. So this is a virus really, we call it really a true camel virus. It spreads to people who are in close contact with camels. And so this would be individuals who are in camel racing or camel farming. It's killed about 858 people, so far 34%. But similarly, the time to onset of infection is short. People end up in the hospital. There are no asymptomatic or few asymptomatic spreaders spreading MERS around, and so that does limit infection. But what we know about SARS-CoV-2, which came and emerged in December 2019, maybe November, in Wuhan, China, closely related to a bat coronavirus. So again, really highlighting the zoonotic potential based on sequence homology. This virus spread quickly, and the median time to symptom onset is 2 to 14 days. And you may not even get symptoms. And that's the one of the many things about this virus when talking about it as being a public health emergency is the quickness of spread and the ability of people to replicate this virus to billions of copies in their nose, but really have relatively no symptoms or no symptoms at all. Maybe you lose your sense of taste and smell, but it's not going to limit you from going to work because, people have to go to work and make money, but you're a vector spreading to vulnerable populations. And we were completely naive because we had never seen SARS-1. And while there are common cold coronaviruses, there's very limited or no protection from those. So SARS-CoV-2 really further highlighted this kind of these ancestral coronaviruses that jump from animals to humans. And I'm showing you a depiction of the natural reservoirs for coronaviruses. And the one I did my PhD in is an alpha corona, a pig virus that can jump between different species. And then SARS-CoV-2, where it came from, we know that it's mostly highly related to bats, whether it jumped from bats to humans directly or whether there is an intermediate host. Research is ongoing. So with SARS-CoV-2, just to then dive into what happens when it enters your body. And so to first enter your body, it has to be spread from an infected person. So when we think about mitigating strategies or decreasing risk of spread, we're talking about mask wearing, social distancing, because we are the vectors. And this virus is spread through aerosol transmission. Droplet size plays a huge role. So virological, epidemiological, and even physical, so like the physics of it, engineering of these particle sizes has gone into really trying to determine, what's an appropriate distance when you're indoors with people and ventilation is poor, what type of social distancing matters. And I think 6 feet is what has come generally out. But of course, that doesn't mean if you're like 6 feet away from somebody that there's no risk. There's obviously still some risk, but you're just trying to decrease the risk with every mitigation strategy as depicted here. So somebody sneezing, these large droplets containing tons of virus, are they dropping to the floor before they reach somebody? And what is your exposure level based on protection? Okay, so let's say you have no protection, you're inhaling this virus because of coughing or sneezing the droplets. What is happening inside the body? And so this coronavirus, as I mentioned, it binds a receptor on the surface of our cells called ACE2, but it's also helped by an enzyme called TMPRSS2. Both of these are required to bind the virus, bring it in, and cleave the spike protein so that this virus can then open up and inject its genetic content into our cells. After the genetic content is inside this vesicle, the RNA is released and translated into protein, so more virions can be made. So we are the replication factories for this virus inside our upper respiratory tract. And so as the virus replicates, increases spread, and infects new cells, it really causes this kind of multi-system response based on where it can replicate. We actually find SARS-CoV-2 RNA in many different parts of our body, so saliva, the nose, the lungs, but even in stool samples as well. And so trying to determine, the ACE2 expression levels, does that relate to the ability of this virus to replicate to higher titers? But then RNA is not all that matters. What about productive virions, productive virus? And really the upper respiratory tract is where this virus is produced most effectively and then spread. So thinking about this picture here and all the different organs where we're finding SARS-CoV-2 RNA, we can then relate it back to how we are affected during virus replication. And this is to do with our immune response to the virus, which I'll discuss in a bit. But some of the impacts of SARS-CoV-2 are kind of wide ranging. So it's not only pneumonia or other damage happening in the lung, acute respiratory distress syndrome, your lung alveoli are responding by producing mucus and fluid, and you're having to have that drained off if you're in the hospital. But it's really a disease of the capillary system. It has a really ability to disrupt the endothelial cells of your blood vessels. And so often when we think about SARS-CoV-2 infection, people with hypertension, with heart congestive issues, with blood circulation issues are really impacted, and that is a risk factor for this disease. But other things, the COVID brain fog, I've heard quite a bit about headache, conjunctivitis, loss of smell. There's some really interesting data on the destruction of the cells that help us taste and smell. And some people aren't able to really taste. It's been months and for some even maybe up to a year, and they're still struggling with food tasting like this burnt taste. It's very interesting. And then other organ system failures if the disease becomes that severe. And so one of the concepts that I like to discuss, and this is a collaboration, Daniel Griffin is an MD at Columbia, and he does a lot of science education on kind of these phases of COVID-19. And I did a blog post if people are interested in sharing this, but it's really been recapitulated quite a bit in the hospital, these phases of COVID-19. So you have a viral phase where the virus is replicating to high titers in your body. And maybe that's during the pre-symptomatic phase where you're not recognizing that your body is secreting huge amounts of this virus. And then by the time you start feeling poor, maybe around day 7, you might end up in the hospital. Likely, if you have severe disease, you do. And you have this cytokine, I crossed out the word storm because immunologists, we actually are, this is not technically a cytokine storm, even though some people are calling it that, but a cytokine phase, which are just communication molecules to tell your body, hey, something is wrong. We need immune cells to traffic to the site of infection to be able to clear infected cells. But cytokines can do quite a bit of damage. And what they do is they signal to your blood vessels to open up, to allow the immune cells to migrate through. But what happens when you signal to the blood vessels to open up is you have vascular leakage. And if you have this unchecked inflammatory response potentiated by cytokines, you're going to have this coagulation phase where your body is responding to this influx of fluids and an influx of vascular leakage. And this is where it gets really problematic. We have no drugs to treat SARS-CoV-2. And so how do you deal with an individual who's now has a pulmonary embolism, and that's end up maybe how they died. Recently, a friend's father died of a pulmonary embolism because of an unchecked cytokine storm phase, induced by virus replication. And so these phases, it becomes a nightmare in the hospital to try to understand, what are we dealing with here? Are we dealing with high cytokines? Are we dealing with vascular leakage and clots in the heart or the lung? And then if we can get past that, there's this late hyperinflammatory phase where up to maybe a month or more, people are having this spike of inflammation, whether it be to recurrent virus that's still not cleared from the body, we're not sure, or just unchecked inflammation. These phases of COVID are really problematic. And I'm showing here a person in the prone position, which really, if you're a severe COVID patient, this is the position you're in for most of your hospital stay. And for an individual who's overweight, which is a risk factor, this is very difficult. You want to avoid becoming, a person who's in this situation. And it really is the cycle of trying to understand what's happening until, sadly, if your lungs and your body just can't handle it anymore. And so this is, for those who are interested, can dive into this paper by Daniel Griffin and colleagues, because this is a more nuanced way of looking at viral replication, which has peaked between days 5 and 7, and then this immune dysregulation phase, and some of these ground glass infiltrates that people will find in the lungs of COVID patients, and how that relates to symptoms. So I recommend you to look at that. But to go back to a more simpler phase and why timing matters, these are some of the drugs that have shown positive benefit for mortality and morbidity. There's only one antiviral that is approved by the FDA. It's called remdesivir, but remdesivir actually interrupts and does a frame shift in the virus's replication strategy. So essentially, it messes up the ability to make more copies and more virions of itself. But the problem is people don't usually end up in the hospital until after the viral phase has peaked. And so you can imagine that an antiviral given too late, by the time the immune response has already started, it's difficult to implement. Now, there has been, I think the clinical trial showed a reduction in hospital stay by 1 to 2 days, which is great financially, but also, trying to get out of the hospital to recover. But it's still a challenge because people are not ending up in the hospital maybe till after the virus is already replicating. Monoclonal antibodies, I'll talk a little bit about antibodies, but these are proteins that give the ability to block the virus. Again, important during the viral phase, although have shown a lot of efficacy because they're very specific to SARS-CoV-2, unlike remdesivir, which is just a general antiviral. Tocilizumab was an anti-cytokine. I've brought up cytokines a lot, antibody to help block maybe some of these pro-inflammatory molecules that has shown some efficacy. But one of the biggest ones is corticosteroids. But the timing of this matters. You do not want to give a corticosteroid that suppresses the immune system early on when you need your body responding to the virus, but you want to give it later on when you're maybe undergoing this severe inflammatory reaction. I do want to touch upon long COVID because it's a really big problem right now. There are, hundreds of thousands of individuals who have survived SARS-CoV-2, but are ending up in this like purgatory of having new or ongoing symptoms, whether they had symptomatic disease or not, but individuals who maybe aren't even able to return to work because shortness of breath, fatigue, cough, headache, all of these symptoms are lasting months and months. And then maybe even more serious is multi-organ failure. And I don't have time to show you, but this was a video given by patient's permission of an individual who has had COVID, has been in the hospital for many months and is on a ventilator because they're for whatever reason, their body has been triggered for this inflammatory response, but now to deal with the destruction, it's very challenging. So this is something that our current medical system is not equipped to deal with people with this much chronic disease. And so it's been something that the NIH has been funding to try to understand. So to quickly, in the next, let's say, a couple minutes, really talk about the innate and adaptive immune response to SARS-CoV-2. So innate is the quick response, adaptive is slower, but very specific. I'm showing this general slide. There's a lot of specifics in here, but basically the innate response, the goal is to quickly trigger something called interferon, which is IFN, and then it stimulates these innate cells to secrete these cytokines that can bring in immune cells. They kill infected cells. Of course, when this is unchecked is when you have a hyperinflammatory response. But at the same time, what coronaviruses are known to do is to limit interferon production. And so what I'm showing down here in C is if you have a normal interferon response, you can contain the viral titer. But if you have a delayed response in elderly individuals and individuals who are immunocompromised, your titer becomes higher and you have greater immunopathology. So innate, the quick response, matters to the immunopathology. The adaptive response, which is what the vaccines are based off of, is talking about B and T cells. These are the cells that produce memory to SARS-CoV-2. They produce cytokines, they produce antibodies that the B cells do. And so after an innate response, these very specialized cells called dendritic cells present the antigen, and you develop memory, so that if you see that again, if you see that spike protein again, your body knows what to do to respond better and higher, with more zest, to produce more antibodies to help prevent infection or prevent the spread. And so adaptive immunity is what we talk about with vaccines. There are multiple different vaccine technologies. Just briefly, there's mRNA, which is a message to produce the spike protein. It's wrapped in a lipid nanoparticle, so there's a fat particle being injected, and the mRNA produces the spike because it has to be picked up by those dendritic cells to present to our body to make antibodies. The Johnson & Johnson is an adenovirus. We are naturally infected with adenoviruses all the time. This technology uses an adenovirus, which would not harm us, to give us the code to produce the spike protein. So just multiple ways of the same thing to show our bodies the spike protein to produce antibodies, as this figure shows. And those antibodies will then bind SARS-CoV-2 to inactivate the virus. And so that's why us as immunologists and virologists, the vaccine is so important, because I've never in my lifetime thought I would see a vaccine with efficacy so high, 90 to 95%. So that is just, it's amazing. And so it's going to be hard for other vaccine viruses to reach the success of SARS-CoV-2 vaccines. Do you want to worry about variants? It's definitely a point to worry. We do follow and track this. Variants are caused by mutations. It doesn't mean that this is a different strain. It just means that these are mutations, typically within the spike, but other parts as well, that emerge when the virus is replicating unchecked. The best way to suppress variant emergence is to stop the replication and of spread of this virus, because every time a virus replicates, and those in the audience that know viruses, it makes tiny mistakes every time. Now, every mistake does not lead to more transmissibility. A mutation could lead to the virus being a dud. It doesn't produce more virions. It could be neutral. It doesn't cause any issues. Or it could evade the immune response, which is what we think is happening, particularly with the Delta variant. But I want to emphasize both my own opinion and the CDC and FDA. There's no evidence to suggest right now that booster vaccines are needed in healthy individuals, and the vaccine-induced immunity is still effective. There are circumstances, immunocompromised people, maybe somebody whose antibody titers are really waning, and that you could talk to your doctor about. But for right now, the recommendation is that a booster would not be needed, and we need to monitor that for sure. Looking towards the future, so as we go into this post-vaccine fall and winter, we're thinking about herd immunity. Dr. Whitmer's presentation was a perfect starter to this concept of herd immunity with plant viruses, but as well as humans and animals. The amount the virus can spread is only dependent on the vectors or the host that it has to replicate inside. And so you can see this depicted here. The Delta variant does make us concerned, but let's see what happens as people start to congregate. And then, do we need to boost the Johnson & Johnson vaccines with Pfizer? We've seen some good data that is effective. And then, of course, monitoring the Delta variant. So I want to leave time for questions, but some of the lessons learned are very similar, again, to Dr. Whitmer's, and really brings home this concept of One Health, which is the slide I'll end my talk with, is that we are vulnerable to viral pandemics, both human pandemics, animals, and plant pandemics. We are vulnerable. We need greater surveillance because these animals are zoonotic. Every mammal that I've ever studied or looked at has a coronavirus, and these, the way that it replicate, can they bind to a human receptor on a human cell? We only know if we know what coronaviruses are out there in bats and pangolins and civets and raccoon dogs. So it's very important because we need investment in these things before they emerge. We had some idea of what drugs worked from SARS-CoV-1, but very, very little. And I think that if we took all the viruses with pandemic potential, plants, animals, and humans, and identified drugs ahead of time, we'd be way ahead of the curve. Pandemic response teams. And then one of the biggest things, and one of the things I'm struggling with right now as a science communicator, is we really need everybody in this effort who are in science to communicate the science to the public. I think I can't spread this information as quickly as social media or a political news show. So we need to come together to try to educate on science, because not only is it exciting, a field that has a lot of job potential, but obviously is important for pandemic responses. And so I'll leave you with that, with hopefully 6 minutes of questions and this concept of One Health and working together. Those of us on the medical side that I'm in now, I was at veterinary school at Ohio State, but then working those with plant viruses as well, because as you know, vectors are not just humans and animals, but also insects. And that plays a huge role. So I want to thank you so much for inviting me. I'd be happy to take questions. You can contact me, email, Twitter, or check out the podcast, because we do talk about a lot of cool things. So I appreciate your time.
Thank you very much, Stephanie, for a great presentation. And we do have time for some questions. And if you'll send those to the Q&A button on the toolbar, I'm going to read those out to Stephanie, and if you could address those. So sure. It is a great talk. This is from Charles Chin. Thank you. Is there any way to predict new variant mutated in possible gene sites so we can develop a universal vaccine?
Yeah, it's a great question. So one of the best ways is surveilling the viruses that are replicating in human populations and starting to see what mutations are popping up. And so that is something that you hear a lot on the news, because it gets picked up of a new variant here, a new variant there. So by studying what are the sites of vulnerability in the virus, and so the spike protein is what we learned is the antigenic site, but there's specific regions in the receptor binding domain that we can target to create this universal vaccine. And there's a DHVI at Duke. There's been, Bart Haynes has published quite a bit on strategies for universal vaccines. And as you'd imagine, the spike protein is where mutations happen often, but then that can lead to evading immunity. And the other strategy that I would propose is to follow the mutations in the nucleocapsid. It's a more conserved region. And so for universal vaccine, I think targeting both an antigenic site that mutates quite a bit, the spike, but also the conserved regions in the nucleocapsid that are very similar between animals and humans, that together, could lead to better universal protection.
Okay, we've got one more question in here. So great talk and nice presentation. Is it the case that the Delta variant spread from India, where it was first detected, to other parts of the world, or could it just be a result of the virus's normal life cycle where at some point it mutates to a new variant?
Yeah, that's a great question. And I think you're really honing in on this concept of, are these viruses really more transmissible? And we're very resistant in virology to call them that, because just because a virus is spreading more, it doesn't necessarily mean its inherent properties make it more transmissible. It could just mean that, as you mentioned, it's a result of the virus's normal life cycle. There happened to be mutation, and then selective pressure caused it then to evade immunity and take hold. So so I would agree with you that it from an evolution perspective, the virus is not developing properties that make it more transmissible every time. It's this normal life cycle where at some point it mutates, and then human behavior determines what happens after that. So yes, I think you're spot on.
Okay, we've got time for just a few more. Here's one from Soraya Bertioli. Fantastic talk. Possibly the main problem with COVID-19 control nowadays is people's refusal to get vaccinated. Science is being crushed. A bit like what happened with the reaction to GMOs. Effective communication is essential. What are the efforts being made to make it more effective, the communication?
Oh, wow. Yeah, this is like the $1 million question. And it really does span political spectrums when we think about science communication, because you're right. GMO is a really good example of some of the battles we're up against with science communication, and how anti-GMO, anti-vaccine sentiment is really across the board in terms of demographics. So, as scientists, we're in the lab, we're constantly writing grants. So we don't often get to do as much science communication as we want, but I think we each do a little bit. So the series of podcasts on Microbe TV, I don't have the only one, there's a bunch of them. I think it's going to take individual efforts or a requirement by the NIH that to get grants, you have to do more SciComm, but of course, who has time? So it's this balance. It's very challenging. And yes, it is. I think at this point I'm on damage control, and if you can get one person vaccinated, that does limit the spread to their family members. And so I would encourage everyone on the call, if you can get like one person vaccinated, you could actually maybe prevent infection from a whole family unit potentially.
Okay, and I think we have time for one more question. And let's see. Thanks for the presentation. How do you know a particular virus has pandemic potential?
Yeah, great question. So how we would determine this is if we pulled coronaviruses from animals and we put them in cell culture. We can engineer cells to express human receptors. So we have cells and mice that express human ACE2, human DPP4, which is the MERS receptor, human sialic acid. You can bring viruses from a bat, and there have been studies done. It's actually quite controversial. It falls in the realm of this like gain of function experiments, which I think is a poor use of wordage, but this type of work is needed to be able to predict, because if I pull a virus from a bat, I put it in cell culture expressing ACE2 and it binds, to me, that's a virus with pandemic potential. I then put it in a mouse that expresses ACE2, and can those mice then transmit this bat corona between each other with the human ACE2 receptor? If that's yes, we're even more nervous about this virus. And so that's how we will know ahead of time, is doing those type of experiments. They will require heavy oversight, of course, but we have the labs in the US, the BSL-4 labs, to do that. And those experiments should be done.